This question is from the chapter Biotechnology, Principles and Processes from class 12th textbook. Question 148, NEET 2023. Main steps in the formation of recombinant DNA are given below. Arrange these steps in the correct sequence. Okay. So there are total 8 steps. It is a complex process and broadly it can be divided into 8 steps. Step number 1 is isolation of DNA. DNA isolation. Whole genome isolation is done. Next one, we need to cut out. We are creating recombinant DNA. That means the vector and our gene of interest. This kind of combination we want. So to remove this gene of interest from DNA, the whole DNA which we have isolated, we will have to do restriction, enzyme, digestion. So once the whole DNA is subjected to restriction, enzyme, digestion, this particular fragment, which is called the gene of interest, is isolated. So we have to isolate that desired fragment. Isolate gene of interest. And then we have to ligate it in the vector. So next step is ligation. Then we have to increase the number of that product, and this is done by amplification process, PCR. And once we have enough DNA to handle, then it will be cloned, or the right term is we transform the host cell with this cloning vector. Transform host cell. Then grow the host cell, large scale growth. Then isolation of the product and post isolation processing, purification of the product. So these are the 8 steps in recombinant DNA technology. Now we will pick out and arrange them in the correct sequence. Insertion of recombinant, okay, I will use a different color now. Insertion of recombinant DNA into the host cell. Insertion into host cell, that is called transformation. So this is A. Next one, cutting of DNA at a specific location by restriction enzyme. Restriction enzyme digestion. This is B. Next, isolation of desired DNA fragment. Desired DNA is gene of interest. Desired interest. So this is C. Last one, amplification of gene of interest. Here they have used the word gene of interest using PCR. So that is over here, PCR, D. So our answer would be B, C, D, A. Let's look at the options now. CBDA, no. BDAC, no. BCDA, yes. Here is our answer. BCDA. CABD, no. So option 3 is the right answer of this question. Thank you so much for watching this video.